Okay, so this is the fifth and final video here for blood, and what we're going to talk about here, and we talked about it a little bit in class already. We're going to talk about blood transfusions, blood typing, and transfusion reactions. So as I mentioned in class, it's important that we type blood, because if we need blood, if we desperately need a blood transfusion, it's important that we know what our blood type is. Okay, so for successful transfusion reactions, we have to type our blood. And what our blood should look like when it's coursing through our blood vessels is, if we look at a blood smear, we can see that each of the red blood cells is separate from its neighbor. It's able to successfully deliver oxygen to working tissues. Well, if we are given a transfusion that is incompatible with our blood type, what can happen is the antibodies that are in our plasma can attack the transfused blood, and it can cause something to occur that's called agglutination. Okay, this is a blood smear where red blood cells have agglutinated, or they've clumped or stuck together, and we can see that occurring in specific locations very well. Okay, and when those red cells stick or agglutinate together, they're unable to successfully deliver oxygen to tissues. They can clog up blood vessels, cause strokes, cause kidney problems, things of that nature. So our blood must agree, so the recipient's blood must agree with the donated blood. In an incompatible transfusion, it can result in fatality. All right, so let's take a look a little closer here about how we type blood. All right, so what we're looking at here is we're looking at some red blood cells, and on the surface of red blood cells, and on the surface of all our cells, we have things called antigens. Okay, so for our red blood cells, the antigens are little glycoproteins, so proteins that are stuck in the cell membrane of the red blood cell, and attached to those proteins are small chain carbohydrates, and what they do is they signify our blood cells as being a particular type. Okay, and we need to know what those glycoproteins are, because if we need a blood transfusion, again, that's going to dictate whether or not we successfully can receive, or we'll reject the transfusion. So let's take a look. So here in the first red blood cell that I have here, we see some little blue circles. Okay, and those little blue circles are signifying a particular glycoprotein on the cells, and those glycoproteins are saying that that particular red blood cell is of type A. So those are type A glycoproteins. Okay, so when we're trying to determine what our blood type is always going to correspond to the types of glycoproteins, or the types of surface antigens we have on our red blood cells. Okay, so this first red blood cell has A antigens, and therefore the blood type is A. This second blood cell has B antigens, and you can see the B antigens here are these little kind of purple triangles. That blood type is type B. What if we have both blue circles and purple triangles? Well, the blood type is AB, and we can see both blue circles and purple triangles on the surface of this red blood cell. What if our blood cells have no A or B surface antigens on them? Well, if that's the case, then we are a blood type called blood type O. All right, and we can see that on blood type O we have neither A nor B surface antigens. Okay, so we must know what our blood type is to determine whether or not we can successfully receive a specific type of blood from a donor. Okay, so what is it about our blood that might reject a donor's blood? Well, antibodies. So we have antibodies in our plasma, and antibodies are these Y-shaped proteins. All right, PROS, that's proteins. So Y-shaped proteins that attack foreign tissue. Okay, so these are going to attack foreign tissue. All right, so depending on what our blood type is, that's going to tell us what antibodies we have in our plasma that will reject incompatible blood. So when we're trying to determine what antibodies are in our plasma, we always have the antibodies that are opposite of our surface antigen. So I'll write that for you here. AB is antibodies, so we always have antibodies for the antigens that we do not have on our cells. Okay? Another way to think about this is the antibodies are always opposite of the antigens. Okay? It's important that you recognize that antigens are stuck on cells and those determine the blood type. Antibodies float around in the plasma. Those are the Y-shaped proteins that can reject incompatible blood. So someone who is of type A, right, they have surface antigen A, and we know that the opposite of A is B, right? So these people floating around in their plasma, they have B antibodies, and you can see here the B antibodies are these kind of purple colored Y-shaped proteins, and they are going to reject any type of blood that has B antigens on them. So the antibodies are always going to attack surface antigens of their own type. Okay? Someone who is type B blood, they have A antibodies in their plasma because the opposite of B is A. And you can see here that in someone who is type B blood, they have A antibodies. And you can see that the shape of the antibody is the same as the shape of the A antigen. So the A antibody will reject cells that are given to the donor that have A antigen on them. Someone who's type AB blood, they have all of the antigens, so they have no antibodies. Okay, so that's, remember, you always have the antibodies are always opposite the antigen, right? So they have all antigens, opposite of all is none, no antibodies. What about someone who's type O? So someone who's type O has neither A or B antigens, so they have no antigens, and the opposite of none is all. So they have all or both antibodies. And you can see here they have both antibodies for the B antigen, and they have antibodies against the A antigen. Okay, so what blood types can these people with different blood types receive? Well, someone who is type A cannot receive blood that is B or AB. Why? Well, because they have these B antibodies in their plasma, and if they're given a blood type who the cells have the B antigens on them, they'll reject that blood. They can safely receive their own type. You can always get your own blood type, and you can always receive type O. Okay, type O is the universal donor, and the reason for that is because we see there are no surface antigens on type O cells. So what that means is there are no antigens for antibodies to attack. Remember, antibodies are the attackers. Okay. Someone who is type B blood cannot safely receive A or AB. Why? Because they have the A antibodies in their plasma, which means they cannot get any blood who has A antigens on them, because remember, the antibodies always attack their corresponding antigens. Someone who is type AB blood, they can receive blood from anyone. Why is that? They are considered the universal recipient. Okay, why is that? Because they have no antibodies. If they have no antibodies, that means that they have no attackers to reject blood they're given. Okay, someone who is type O, they can only receive blood type O. Why can't they receive A or B? Because they have antibodies against both the B antigen and the A antigen. Okay, so they cannot get any blood that has A or B antigen on it. Okay, they are the universal donor, and again, that is because they have no antigens on the surface of their cells, and if you have no antigens on the surface of your cells, then no antibodies will ever attack and reject that blood. Okay, let's move forward. There's one additional antigen that we need to become familiar with, and that is an antigen called the D antigen, or the Rh antigen. And that tells us whether or not we have a positive or a negative blood type. So a positive blood type indicates that we have the D/Rh antigen. Okay, and I told you in class that the D antigen and the Rh antigen or Rh factor, they mean the same thing. Okay, so if we have a positive blood type, it means we have the D antigen on the cell. Okay. If we have a negative blood type, that means that we have no D/, not or. We have no D/Rh antigen on our cells. So if we're a positive blood type, it means we have the presence, or that D antigen is on the cells. If we're a negative blood type, we do not have it. Okay, so I gave you an example in class. Okay, if we have a cell who doesn't have any A or B antigens on it, it's an O blood type, right? But if it has some D antigens on it, this is a positive blood type, right? So this would be O positive. Okay, let's take a look at another O positive blood type. All right, if we have B surface antigens and we have D surface antigens, this tells us that the blood type is B positive. Okay, if we have just A surface antigens on a cell, this indicates that the blood type is A negative. Why is it negative? Because we have no D antigens. Okay, no D antigens, it's a negative blood type. If we have a D antigen, it's a positive blood type. Okay, so you always need to indicate when you're giving a blood type the AB antigens and the D antigen if it's present. Okay. All right, so let's move forward and talk about blood typing. So we talked about this in class, but I just want to reiterate it here. So when you're blood typing, we're given some samples, and what we're trying to do is we're trying to figure out what antigens are on the cells, the red cells in that sample. Okay, there may be some antigens, there may not be, but we need to figure out if there are, and if in fact there are, which ones. Okay. So how are we going to do this? Well, we can take some sample, and let's imagine that we're going to take sample 1, and we're going to put sample 1 onto a well plate. All right, so here's 3 drops of sample 1 into 3 different wells on a well plate. Okay, and now what we can do is we can use some antibodies to check to see what the surface antigens are. Okay, we know that if we mix antibody with cells that have the antigens on it, that antibody can attack, that we'll get this process called agglutination. Okay, so if I add some antibody D to this well here, and I stir that sample up, if the sample causes agglutination, if that sample agglutinates when I add anti-D to it, this is telling me this is a positive reaction. But what it's really telling me is that there is the D antigen present. Okay, so we can use antibody to check for antigens, because we know if we add an antibody and we get a reaction, that means that whatever antibody I'm using, the antigen is present on those cells. Okay, so in this case it would be, if we got a reaction, this would be a positive blood type. All right. Let's assume that I add some anti-A, so some antibody A, to this well here. All right. And I stir it up, and I also get a reaction. So I also get agglutination. So I got agglutination telling me that the D was present, and then I added it to a different well, and I also got agglutination. What does that tell me? Since I'm adding antibody A to the sample, I'm checking for the presence of the A antigen. And if I get a reaction, that tells me that the A antigen is present. And if the A antigen is present, well, then I know that I have a positive blood type and I have the A antigen also present. Okay, so I know that my cells in that sample so far, they look like this. They have the D antigen on it and they have the A antigen on it. Now I still have to check for B, and I can add the anti-B to this final well, and I can stir it up. And if I get a negative reaction, so this is a negative reaction, I got no agglutination, that tells me that there is no B antigen on the cells. So I can safely say that my cells in sample 1, they have both the A antigen and the D antigen. And if we have the A and the D, what is the blood type? A positive. Okay? All right. Finally, one last thing I want to talk about here is a condition known as erythroblastosis fetalis. All right, it's also known as hemolytic disease of the newborn, or HDN. Okay, so how does this work? This is a condition where we have a mother who is Rh negative. Okay, so mom is, we'll call her D negative, meaning her red blood cells have no D antigen on them. And we have a father who is D positive, meaning his red blood cells have the D antigen. Okay, so in this condition, in order for this condition to occur, mom has to be D negative, okay? All right, so what happens? So mom and dad have a child, okay? And we can see that in this pregnancy picture here, this is mom right here. And all these negatives indicate that mom is D negative, okay? However, mom's baby is D positive, and that is indicated by these positives. So mom has a negative blood type, baby has a positive blood type, because baby got the gene for the positive blood type from dad, because dad was positive, so he passed the positive gene on to baby. Okay, so let's take a look at what happens. All right, so if we come up to this little drawing that I have up here, we can see that this is a pregnancy. It's the first pregnancy, and this is mom's blood type on the left, and this is baby's blood type on the right. And we know that mom is D negative. Okay, so mom has a negative blood type and baby has a positive blood type. Okay, and the D antigens are represented on the baby cells as these little kind of molecules on the red blood cells of the baby. Okay, so in order for this condition to occur, all right, during the pregnancy and during the birth, the baby's red blood cells somehow get exposed to mom's immune system. Okay? They cross over and they get exposed to mom's immune system. When is this going to occur? This may occur during the birth. Okay? So normally throughout the pregnancy, all nutrients are passed from the mother or from the baby to the mother through diffusion. There's no direct contact of the baby's blood with the mother's blood, or of the mother's blood with the baby. However, during birth, if the baby's blood cells get into mom's circulation, okay, mom will see these babies, the D antigen. Mom will see that D antigen as being a foreign antigen that she has never seen before because she doesn't have the antigen. Okay? As a result of seeing that D antigen and thinking it's foreign, mom's immune system can start making antibodies against the antigen. She doesn't have the antibodies to begin with, but she can start to make them. Okay? And if mom sees that D antigen, mom's immune system can become sensitized. And what that means is that mom's immune system will start to make D antibodies. She didn't have them before this happened, okay? The first baby gets born and the first baby is normal. So we can see that here. Baby number 1 is normal because the mom was exposed to the baby's blood during the birthing process, okay? And what that shows is over here in picture C, this is what we see. So the baby's red blood cells get into mom's system, and mom sees those positive D antigens as foreign. Baby is born, baby's fine. However, we can see in picture D, mom now has made D antibodies. So she has D antibodies now flowing through her blood and in her system. If mom gets pregnant again, baby number 2, pregnancy number 2 occurs and mom is still negative, and the second baby is also positive. So this is baby number 2. Baby number 2 is positive. We can see baby number 2 has the D antigen just like the previous baby. Mom's immune system has produced these D antigens, or these D antibodies rather. So now these D antibodies can cross over the placenta. So this is the kind of placental barrier. Those D antibodies can cross over the placenta and go and attack the baby number 2's red blood cells. Okay? And if that occurs, what can happen is we can get agglutination. Okay? That baby number 2's blood will start clumping up and agglutinating. And this condition is known as hemolytic disease of the newborn. Those red blood cells will cluster up and they'll start breaking open. And hemoglobin will start floating through the cardiovascular system of the baby and will get stuck in the kidneys and will cause severe anemias, and the baby can die. Okay, so that condition can occur if mom has D antibodies in her system and she is pregnant with a baby who has a positive blood type. Okay, how can we prevent this from happening? Well, if we know what the dad's blood type is and we know what the mom's blood type is, just to make sure that this does not occur during the first pregnancy, mom does not become sensitized with a baby who is positive, what we can do is during that birthing process, we can give mom a shot of something called RhoGAM. And what RhoGAM does is it prevents mom's immune system from making D antibodies. So it kind of stops her immune system from making D antibodies, even if she does get exposed to baby number 1's D antigen. Okay? And that way, during pregnancy 2, even if the baby is positive, mom won't have those D antibodies because we gave her the shot of the RhoGAM that prevented her from making those D antibodies. Okay? All right, so that's the end of the blood typing screencast.